The purpose of this video is to walk you through how mutations occur and what happens when they occur. And in order to understand how that works, you need to have a pretty firm understanding of protein synthesis. Because if the protein is made wrong, that's where mutations really make their name in an organism. So here's a ribosome going along, reading a piece of mRNA, bringing in tRNAs, and most importantly, bringing in amino acids. It's that sequence of amino acids that could potentially, if placed in there incorrectly, could potentially result in a mutated protein, which could affect an organism for the better or for the worse. Something to keep in mind, that mutation doesn't mean bad. Mutation could mean good just as well. And so I'm going to take this picture right here and put it into our presentations here. So here's a picture of the ribosome going along that mRNA, reading the mRNA, bringing in tRNAs, and then most importantly, the amino acids. So here we have amino acids being linked together because the code that was given to the mRNA by the DNA. And an important thing to recognize is that the anti-codon of tRNA is always going to line up, hopefully correctly, with the mRNA codon. And to see kind of how that works with mutations, or how to decide which codon or which amino acid is supposed to be brought in, we have this thing called the codon chart that you can see here. And on this codon chart are all the amino acids necessary to produce life, at least in your body, to make things work in your body. All right, and how this works, you can see here it says first base, second base, third base. This isn't talking about baseball. We're talking about the bases on the codons, right? And again, every three set of letters here is a codon that's going to arrange themselves, hopefully correctly, with an anti-codon and bring in the correct amino acid. Again, mutations come up when the wrong amino acid gets brought in, or an amino acid does not get brought in, things like that. So the codons that we're talking about for this codon chart, again, are on the mRNA. The codon chart does not have to do with the tRNA. This is the complementary side to the tRNA anticodon. Right? So the mRNA codon makes up your codon chart. And when it says first base, second base, third base, it's just talking about the letters within each codon. So here's AUG, first base, second base, third base. The next codon would be CCG, first base, second base, third base again. So we're going to use this picture to determine how these amino acids were brought in correctly and line that up with our codon chart just so you can see how this all works. All right, so like I said, our first codon here is AUG, if you can't see it. It says AUG right there. So let's use our codon chart to determine, if it wasn't already on here, to determine which amino acid should be brought in if the codon is AUG. So again, we go to our first base here, and our first base is an A. So we travel down here till we find our A. Our second base is a U. So we're in this box right here. All right? And if you see all the way on the right side, here's our third base. In each row, we have UCAG, UCAG, UCAG. Well, we're looking for the G. Our third base is a G. All right? But it has to be in this row and column, because that's where we've decided from these first two where to begin looking for that last base. And again, it's a G, which is the last one. So right there, methionine. And you'll also notice that it says start here. This is the starting amino acid for all proteins. Okay? So you'll always see a methionine at the beginning of each amino acid chain. Right there's methionine, because that is always the first amino acid in a protein. Next codon is CCG here. Right here is CCG. So again, let's see where that proline came from. In our codon chart, CCG, again, you're looking at your, oops, didn't mean to do that. CCG, we're looking for the first base being a C, the second base being a C, the last base, the third base being a G. So here's C, C, and again, the order in each box is UCAG. Not that it matters here, because all of these code for proline. So no matter what our third base is, this next amino acid is going to be proline, and that turns out to be correct right there. PRO, abbreviated PRO, just the first 3 letters typically. So for the last one that is coming in, we see UAC here. Right here, this codon is UAC. So again, one more time through our codon chart. Let's see how that works out. UAC, so U is our first base. Okay, A is our second base. So we're still in the same spot. But C is our second letter. Oops, sorry, I messed that up. U, A, C. That says tyrosine, which is correct. TYR is the amino acid that's coming in. Where problems can arise is if this mRNA is made incorrectly from the DNA, or the wrong tRNA gets brought in, also bringing in the wrong amino acid. Or if bases are missing or inserted. So that's how to read a codon chart. If you can't read this, it's hard to understand why the protein is made correctly or incorrectly in the case of a mutation.